Hello and welcome to our discussion on hormone receptors and mechanisms of action. So we already know that our hormones can be broken down into two classes, a water-soluble hormone classification and a lipid-soluble hormone classification. So our water-soluble hormones are going to fall into this area where they're going to attach to the membrane-bound receptors. Now those receptors are going to either alter the activity of G proteins, or we're going to have receptors that will alter the activity of intracellular enzymes. If we go this route, we will activate existing enzymes and get the cell to respond. If we go the G protein route, then we can open or close the ion channel depending on what we need to do and activate existing enzymes that way, which will lead to the cell responding. Now, if we have a lipid-soluble hormone, then we will be able to attach to nuclear receptors within the nucleus that will activate genes, synthesize new proteins or enzymes, which will allow the cell to respond. So this is a good slide to come back to once we've finished this part of the screencast. So let's talk about target tissue specificity and the response. The portion of our molecule where the hormone is going to bind to is called the binding site. So in this image here, we could see that our hormones are circulating, and hormone number 1 has a specific receptor on its target cell for hormone 1. Whereas this other hormone that's traveling through, if it attempts to leave the capillary and tries to bind to this hormone receptor, it cannot, because it is not the correct conformation to match this specific receptor. And this is how we're going to get specific cell responses. So this would be called our receptor site at this point. The example that your book gives is the epinephrine hormone cannot bind to the receptor site for insulin. Although I will mention that there are a lot of cells that have multiple receptors on them. So they may have epinephrine acting on one cell and have a receptor site on there for, let's say, a thyroid hormone. All right, we need to continue on to talk about upregulation and downregulation. We can change the number of receptors on our cell. So let's first talk about upregulation. This is when we are able to increase the number of receptors on our cell. So both of these cells on the right and left of your screen have about the same number of receptors. So if we get a hormone that comes over, goes inside our cell, and through its cellular response, we can form more receptors. If we have downregulation, our hormone can enter into the cell and cause a decrease or desensitization of our receptors here. So now let's talk about mechanisms of hormone action. Through hormone action on our target cells, we will be able to do the following. We can alter the plasma membrane permeability of membrane potential by opening and closing ion channels. We can stimulate synthesis of proteins or regulatory molecules. We can activate or deactivate enzyme systems. We can induce secretory activity and or stimulate mitosis. So we're going to take a look at some examples of these as we discuss the endocrine system further. So first, let's talk about our classes of receptors and the hormones that will be attaching to them. So we'll start off with our lipid-soluble hormones that are going to be able to move through the plasma membrane with ease and attach to nuclear receptors that will directly activate genes. So our hormones that can do this, like we already mentioned, would be lipid soluble hormones, but we can also have smaller molecules do this as well. They are able to react with enzymes in the cytoplasm, or they can react with DNA to cause transcription and translation. Remember that transcription would be creating that mRNA strand, and translation would be the reading of that mRNA strand to create a protein. The possible examples that we will talk about here would be your thyroid hormones, your testosterone, estrogen, progesterone, aldosterone, and cortisol. All of these that we have already mentioned that are lipid soluble hormones. This can also take some time in order to get a cellular response because we do have a latent period of several hours because of the time that it's going to take in order to go through transcription, create that mRNA strand, and also for translation to take place and create the protein. So now we can talk about our water-soluble hormones again. They are going to bind to the membrane-bound receptor on the plasma membrane. These are called integral proteins because they have integrated themselves into the plasma membrane. And these water-soluble hormones cannot just move through the plasma membrane. They always have to attach to a membrane-bound receptor. Now, when they attach, they are going to be coupled with this G protein complex that is made up of 3 subunits. And this will send over the one of the parts of the G protein, which we'll get into in a bit, over to adenylate cyclase. That will convert ATP over to cyclic AMP that will activate protein kinase in order to create the cellular responses, or it starts the cascade, the enzyme cascade here. So the hormones that will always need a membrane-bound receptor are, of course, our water-soluble hormones, but they can also be hormones that are larger. So you've seen that for our hormones that have to bind to the membrane-bound receptors include intracellular mediators. These mediators are going to be ions or molecules that enter the cell or are produced in the cell. And they could be produced because of G protein activation. So let's take a look at this table over here. There are quite a few intracellular mediators, but we are not going to dive into the ones that work within that are examples of our smooth muscle cell type. Instead, we will focus on the cyclic guanine monophosphate, or cGMP, that is an example of kidney cells, and they are going to work to increase sodium and water excretion by our kidneys. And the other one we will focus on is cyclic AMP that we just spoke about,
Cyclic AMP,
or cyclic adenosine monophosphate, which an example of that cell type would be the liver cells. So the example of the response would be increased breakdown of glycogen and release of glucose into the circulatory system. Okay, so in this diagram, we can see a water-soluble hormone has bound over to the membrane receptor. And as it does this, it activates the G protein. So we said this was made up of 3 subunits. We have the gamma, beta, and alpha. So you can see here that the alpha subunit has left the beta and gamma subunits moving through the plasma membrane over to adenylate cyclase. So at this point, it has activated adenylate cyclase so that it can convert ATP over to our cyclic AMP. We say that our cyclic AMP is our second messenger and the hormone is our first messenger. So what does our second messenger do? It is going to activate our protein kinase enzyme, which will go through and phosphorylate specific enzymes, activating them to cause our cellular responses. The other thing that is going to do is use the enzyme phosphodiesterase to inactivate the cyclic AMP by converting cyclic AMP to AMP. It's in active form. So now let's take a look at signal amplification. What this means is when our water-soluble hormone attaches to a receptor, it's activating G proteins. And when it does this, it amplifies all the other enzymes or signals. So we have even more activated adenylate cyclase, more activated cyclic AMP, and even more activated protein kinase enzymes for the cellular responses to take.
Mama, I'll tell something now.
Okay, see.
Bacteria could drill in your teeth, and your teeth need you need to brush your teeth very good when you eat chocolate or lollipops. You need to brush your teeth very good.
And what else?
And floss every day.
That's right.
I can pick up that again.
Is that it?
And floss every day.
Okay.
And then don't let water come in.
Yeah.
And
that separates your outer ear from your middle ear, right?
yeah, right. That separates the eardrum separates that from your ear to that ear.
Yeah, the outer ear from the middle ear.
Yeah.
Okay.